Hi everyone, this is Sumathi, Zoology Department. Today we have known about the subject. Second we is Zoology student, the subject is Cell Biology. Unit also, third unit. This unit also very interesting. Why? Because all the cell organelles also we have to know about it. The cell organelles mean not only human cell center we have the nucleus and the nucleolus and so many cell organelles. One of the cell organelles is also Golgi apparatus. The Golgi apparatus is also one of the very important cell organelles. It also cytoskeleton in the cell. Normally the Golgi apparatus is multiple names, Golgi complex, Golgi bodies, everything also. Why we called Golgi? The scientists also identifying these Golgi apparatus bodies. So the discovered organelles also Camellia Golgi. The scientist name also Golgi. So we called the Golgi apparatus. It is found in all eukaryotic cells, plant cells as well as animal cells also. You know the difference between prokaryotic cells and eukaryotic cells. In prokaryotic cells, we have that means we have not a true nucleus. In eukaryotic cells, only have the true nucleus. They are membrane-bound organelles, and also it is present in the cystole of the cells. And let us explore more about Golgi complex also. This Golgi complex also gives the shape of the cells, and it has so many cell organelles and the cell structures also. It has the cisternae phase and a lumen-like structure. Namely, it forms a vesicle-like structure and a tubule-like structure. And the lumen-like structures also normally tube-like structure of the Golgi apparatus. Why it is called? Why it is important for the new forming of the vesicle and the secretory vesicles also? It is filled with some fluid. It gives support to all the cell organelles. So it is transporting the incoming of the vesicles and the transferase. It acts as a protein synthesis also. Normally, the Golgi bodies is comprised 5 to 8 cup-shaped series of compartments that is called the cisternae. This cisternae is a flattened shape, and sometimes it may be disc shape, sometimes it may be stacked pouches and a makeup of the Golgi apparatus. Normally, the Golgi stack mostly contain 4 to 8 cisternae. However, it is less than 60 cisternae is found in the protist order of the animals. Normal mammalian animal, all eukaryotic animal cells contain 40 and 100 stacks of cisternae. The animal cells generally contain around 10 to 20 Golgi stacks as per cell, which are connected by tubular connection. And it also Golgi complex is mostly found near the nucleus, and it gives the nucleus and all the cell organelles is connecting the endoplasmic reticulum also. It gives the shape also. So the Golgi apparatus is also very important. And these Golgi apparatus is a creation and the evolutions, whichever one, it believe worked in the wondrous way to evolve the design of various living organisms in the world. Mostly optimum ways it gives the support of the cell. For example, the Golgi complex has been designed such a way it ensures sufficient number of Golgi bodies are present in the cell as per the requirement. After that, we can do all the works normally in day-to-day activities. So the Golgi complex also a very important way to ensure the Golgi bodies in the cells. What are the function of Golgi bodies? The main function of Golgi bodies is a packaging and the secretion of the proteins. Proteins also very important, you know. Why the protein also important? Protein also bodybuilder. The combine of the amino acid formation of protein, after that protein, our body also activates a normal body function. It receives the protein from endoplasmic reticulum. Already I told you, during protein synthesis in the endoplasmic reticulum also very important. Normally endoplasmic, two type of endoplasmic reticulum occurs. One is a rough endoplasmic reticulum, another one is a smooth endoplasmic reticulum. It depends upon the presence of ribosome in the endoplasmic reticulum. It may be vary. Ribosome is present in the endoplasmic reticulum. It is called a rough endoplasmic reticulum. It is a package of some membrane-bound vesicles, and then it is transported various destinations. What are the various destinations of cell organelles such as lysosome, plasma membrane, and secretion? They take the part of the transport of lipids and the formation of some lysosome. What is the function of lysosome? Lysosome function is a lytic function. Lytic, any one of the particles, foreign particles enter to the body like bacteria, virus, it also dissociated and digested by the lytic enzymes. After that, they have to eject them. Normal post-transitional modification of enzymatic process occurs near the membrane surface, that is called Golgi bodies. Example, phosphorylation and glycosylation in so many bodies, etc. Normal Golgi apparatus is the site, the synthesis of the various glycolipids and some glycomycins also. Normal plant cells, the complex polysaccharides of the cell wall are synthesized in the Golgi apparatus. What is going to be polysaccharides? Many saccharides. Poly means many. Saccharides also occurs in the Golgi apparatus. What is a function is over in the Golgi bodies. After that, we have to know about the centrioles. It is one of the important cell organelle. The centrioles is an organelle and its shape is a cylindrical shape. It is composed of protein. That protein is called tubulin. All the part also, or body also made up of protein. Each and every protein is also named. Depends upon the places the protein and the amino acids also secreted. After the formation of protein, all the animal cells have the two centrioles. They help to cell during the cell divisions is also important. They work during process of mitosis and meiosis. Mitosis is a cell division. Meiosis is a reduction division. They could be found in some lower plants such as Chlamydomonas, although they are not present in the many of the fungi. Angiosperm. The normally angiosperm is always called the flowering plant and the Phainophyta is called conifers. And they are usually present near the nucleus but are not visible when the cell is not dividing. So the centrioles also and distal appendages also occur. The mother centrioles and daughter centrioles we have to divide. What are the difference between these one? Subdistal appendages occurs in the mother centriole. Normally, microtubule triplet is present in the daughter centriole, and also it has the proximal end and the interconnecting fiber between the cell organelle, and subdistal appendages also occur. These distal appendages also occurs in the mother centrioles also. Who discovered the centriole? Bovary and Edward Van. In animal cell, they have to identifying the centriole. After that, they have to know about what are the functions occurs in the centriole. All the centrioles are formed 9 group of microtubules and a triplet. It organizes the cylindrical shape. The detailed structure of centrioles can be studied only under, we can see only through the electron microscope. We cannot see the normal microscope. We are associated together the right angle to each other. When how they identify which animal they have to choose to identifying the centriole, the embryo of the Drosophila melanogaster and Drosophila exceptions and organizations also. The Drosophila melanogaster is a fruit fly. They identify easily to the centriole. The former forms 9 pair instead of microtubule triplet, whereas the premature embryos and the sperm of the elegans have the chromosomes 9 single microtubules also occur. After that, the scientists observed identified the centriole the first time in 1883. Another one is 1888 also. The structure of publication of the centriole was first given by the Joseph. After that, they have to named the centriole helped to organizing the during the spindle formation during mitosis stage. It also completed the process of cytokinesis. Cyto means cell. During cytokinesis stage, the centriole formation is very important. However, the centriole were believed to be necessary for the formation of the mitotic spindle in the animal cell. Although several recent type of research have been explained that the cell which does not have the centriole, in case we can remove through the surgically, can function without the G1 level. You know the cell stages of the cell cycle, G1 phase, G2 phase, S phase, that is a synthetic phase. Everything also occur. Each and every phases also have the some time duration. The G1 level of interface can be formed later to de novo, that is a manner of the cells. Why? Because it will be affecting the mitotic divisions of the cell. Without centriole, it is very difficult to cell division. The location of the centrioles plays a key role in the three-dimensional organization of the cell, and it also regulating the location of the nucleus. In flagellated and ciliated organisms, the location of such organelles is decided after the mother centriole that form of the base of the organism. First, we have to know about what are the role of centriole. After that, we know about the function of the centriole. In spite of being the divide of DNA, the centrioles are capable of forming the new centriole, and it can be transferred into the basal body. What is the role of basal body? To formation of flagella and the cilia. This basal body also very important. At that time also centriole is present in the center part. The cilia and flagella used for movement of the animal from one place to another place. Invertebrate animal. So it helps to cell division, the formation of centriole, to forming from the microtubule to the organism in the center. After that, the centriole, the role of the centriole is formation of cilia in whole the body. These without the centriole, without the flagella, without cilia, the animal does not move from one place to another place. So out of the two centriole, the distal centriole is formed the tail region, another one is the axal filament also. Normal microfilaments and the protein filaments, everything also made up of cytoplasm in the eukaryotic cell and help to maintain the cell structure to perform many other functions. These functions also known as actin filament. Actin is one of the protein, and also it is a part of the cell of cytoskeleton. It gives the definite structure of the cell. After that, if every organelle were eliminated from the cell, the plasma membrane and the cytoplasm would not be one of the component of left. There would be with organic molecules and ions also present in the cytoplasm. The formation of along the network of protein fibers in the cell, it also assists and maintaining the shape of the cell. So it is very important of the microfilament present in the cytoplasm. All the cell organelles also present in the cytoplasm. These fibers occurs at an organelle and a particular position. It allows the vesicle of the cytoplasm and move in the cells and unicellular. It also moves freely from the cytoplasm. The network of protein fibers is called the cytoskeleton. Why? Because the cytoskeleton is important. How it formation of our body skeleton also very important. At the same time, the cytoskeleton is important in all the cells. These exist. Three kind of fibers also occurs in the cytoskeleton. One is the intermediate filament, microfilament, and microtubules. This microfilament is the most narrow fibers occurs in all the cell organelles also. After that, the microfilament is also called the actin filament, as it is consist of the two intertwined. This intertwined strand is called the globular protein, is known as actin. All the amino acids also producing the protein. These actin protein also a formation of microfilament. It also have the one of the structure that is called the polymers of the protein, and its smallest filament is called the cytoskeleton. The vital role of the cell, that is a microfilament, is a movement, cell division. Another one is a muscle contraction and relaxation. These microfilament is provide the shape and the rigidity of the cell. It can be disassembled to reuniform rapidly. Hence, it enables a cell to modify the shape and move the cell. These white blood cells utilize the ability to good extend and good reach the site to infection and the submerge of the pathogens also. Normal microfilament are the leanest filament of the cytoskeleton. It is present in the cytoplasm of the eukaryotic cell with a diameter about 5 to 8 nanometers. We can calculating the meters, nanometers, very millimicron. These polymers of this filament is flexible but very strong and resist the working of the crushing while offering support to the cell. The microfilaments are versatile component and play a major role in the cytokinesis and the shape of the cell. The flexible arrangement of the filament is a framework. It enables to help in the cell movement from one place to another place. The filamentous also play the vital role to contracting the molecular nerves and driven by the one of the protein that is called actomyosin. The actomyosin is a process. The lean filament becomes expanding the platform and pulling the action, it pushing the action is a myosin. It occurs mainly during the pseudopod and develops the contraction. After that, normally we do the functions in the hand raising and hand dropping. We have to use this type of proteins. Next, cell organelles, we have to know about a very important and peculiar cell organelle called nucleus. Where it is present in the nucleus. The nucleus is present in the center of the cell. It is also very, very important. After we have to know about all the cell organelles, we given the importance of the nucleus. Why? Because the nucleus also have the chromosome. The chromosomes also have the DNA. The DNA also have the nucleotide. All the nucleotide also control the gene and our body activity. So this nucleus also very important. The nucleus have the small pore and it is covered by the nuclear membrane and also covering the nuclear envelope also. Centrally, the nucleus have the nucleolus. The nucleolus is surrounded by the endoplasmic reticulum. The endoplasmic reticulum is joining with the nucleus from one side and also it has a thread-like structure that is called the chromatin material or chromosomes. Nucleus also have the small pore in between the cells that is called nuclear pore and nucleolus, and the ribosomes also attached in the endoplasmic reticulum. In surrounding membrane also contain the endoplasmic reticulum. The most integral component of the cell, the nucleus, normally plural we have uses of the nuclei. It is derived from the Latin word kernel of nut. The kernel of nut means nucleus. A nucleus is defined as a double membrane eukaryotic cell organelle that contain a genetic material also. Already I told you that genetic material called DNA. Why? Because it is called genetic material DNA. The DNA is used for transfer the genetic material from one generation to another generation. What are the structure of nucleus is most evident organelles of the cell. It is completely bounded by the membrane. It is engrilled by the structure defined as the nuclear envelope. The membrane is distinguished the cytoplasm and also it contains the nucleolus and the nucleus, everything also. These nucleolus also have the genetic thread-like structures that is called chromosomes. The chromosomes also confined the DNA present of the chromosome. It provides the genetic information required the creation of the different type of cells and the addition of the cells and the reproduction of the cell, everything also based on the nucleus. Already I told you it contains cells hereditary information and control the cell growth and the reproduction. Normally the nucleus has been clearly explained as the membrane. That the membrane bound structure is comprised of genetic material of the cell. It is not just a storage compartment of the DNA, but also it happens the home of some important cellular process in the nucleus. Why? Because it is a cellular process. All the genetic material and nucleotide, everything also controlled by the nucleus, and the nucleus also secreted one of the special peculiar nucleus. So it is not only storing the DNA, but also it controlled all the activities of the DNA. The first and foremost, it is possible the duplicate once of DNA in the nucleus. The process has been named DNA replication, and it produced the identical copy of the DNA. Normally, the DNA is a double helix structure and ladder-like structures also. Adenine, guanine, thymine, and cytosine. Adenine is always binding with thymine. Cytosine is always binding with the triple bond in the guanine. And the producing the two identical copies of body are post the first steps of the cell division. Every new cells will get its own set of instructions. Why? Because it is important. Each and every cells, all the activities also controlled by the genes. The genes also occurs in the nucleus. For example, our heart muscles also have a different function. Our lungs muscles also doing different function. Our eye muscles also have the different visions of the function. So each and every cells have the peculiar genetic information and the secretion of protein to formation of cell. They have to identify coding of the protein, everything controlled by the gene. Secondly, the nucleus is the site of transcription. Transcription also creating different type of RNA from the DNA. You know, transcription and translation. DNA to RNA, RNA is converted into protein. This transcription, the translation would be lot of creating copies to individual pages of human body. It gives the instruction and being removed from the read and the rest of the cell. Normally, the central rule of the biology, the state, the DNA is copied into the RNA and into formation of protein. After that, formation of all the cell organelles. This is the beautiful structure of chromosomes. First, left side it has the chromatin. That chromatin is present in the nucleus, and nucleus also has a nuclear core, and the secondary chromatin and the nucleus of the DNA, and the DNA is a double helix structure. And the normal chromosome also have telomeres, and the arms also have. The chromosome center it also have the centromere. Normally, secondary construction, satellite DNA and the centromeres, everything also present in the external structure of chromosome, and the chromatid and the chromonimeta also present, and helical structures also present in the internal structure of chromosomes. Totally, we have the 23 pair of chromosome in human being, 22 pair of body chromosome, and 1 pair of sex chromosome. Normal chromosome is a thread-like structure present in the nucleus, which carries the genetic information from one generation to another generation also, and it also plays a vital role in the cell division, hereditary, variation, some mutation, and repair and generation also. Normally, the chromosome means chromo means colored body, and the staining ability to the certain dyes also. Carl Nageli identified the chromosome. W. Waldeyer is coined the name chromosome. Now, already I told you, one of the scientists identifying the one cell organelle. Some scientists also named the cell organelles. At the same, what Theodore and Bovary also suggested, the chromosome is a physical carrier of the gene in the all eukaryotic cells. The number of chromosomes in any species is constant in all the cells. The number of chromosomes and gametes, everything also results in the formation of sexual reproduction, and number of preserved in the mitotic division and cell growing repair, everything also based on the chromosome. The chromosome number also varies in the different species. In Nematoda and the Protozoan, Porifera, nematodes, everything also varied. In human being also have the 23 pair of chromosome, 22 pair of autosome and 1 pair of sex chromosomes. The chromatin is readily sustainable in the cell nucleus, and also it consists of DNA and RNA. Normally, we have to differentiate the chromatin. The chromosome is made up of chromatin. Normal chromatin is made up of DNA and the RNA and the protein. The darkly stained condensed region of the chromatin is known as heterochromatin. And the euchromatin also occurs, that is a light stained region of the chromosome, it is called the euchromatin. There are 2 types of chromatin occur. And also it proposes a chromosomal material, the thin filament is known as chromonimeta. Everything also bead-like structure. It is a visible and accumulates the chromatin, the material that is called the chromosome. This is the structure of chromosome based on the number of centromere. We have to differentiate telocentric, acrocentric, submetacentric, and metacentric. This metacentric also has a 2 arms. One is a P arm and one is a Q arm. P arm is look like a short in nature. Q arm is look like a long in nature. And also, these monocentric have the only one. Mono means one. Hollow means it is a centromere have the length of the chromosome. Acentric means the break and fuse together to form a chromosome without centromere. There is no centromere, that is called acentric. And dicentric also, it is a chromosomal aberration occurs during the mutation. The centromere is unstable, the stored centromere. It goes to migrate from the opposite pole of the result, and that is a result of the formation of fragmentation. and telometric, telocentric, acrocentromic, and submetacentric and metacentric. And telocentric, the rod-like chromosomes, already I told you, T-arm also present. And the acrocentric, it is a rod-like structure and it is present one giving rise to a short except in the long term. Submetacentric also L-shaped or J-shaped. Metacentric also V-shaped chromosome, and also it has the two equal arms present. Two types of chromosomes broadly occur. One is a gain chromosome, another one is a Lamb-Rush chromosome. The gain chromosome is also called the polytene chromosome. The polytene chromosome is first identified by Balbiani. These chromosomes first identified in the Drosophila melanogaster in the salivary gland also. It is alternating dark band also occurs. It is a puff-like structure, so that is called the Balbiani ring. It is first identified in the salamander in the Lamb-Rush chromosome. Normally, the Lamb-Rush chromosome found in the spermatocyte. It has a lateral lobe-like structure and DNA symmetrical structure, and the intensity of the RNA also present. This is a structure of chromosomes. Maternal chromosome, another one is a paternal chromosome. Maternal chromosome is a mother chromosome. Paternal chromosome is a male chromosome. This chromosome have the chromosome lobe-like structure. It enlarging the section of chromosome, chromatin lobe, chromomere, chromatid, everything also present in the chromosome. What is the main role of functions? Already I told you important function is acting in the guiding. That is a growth, reproduction, repair, everything also. The chromosome protects the DNA getting during the predatory material transfer from one place to one generation to another generation, and the spindle fiber attached to the centromere also very important. Each chromosome contains thousands of genes, that's very important. It precisely codes the multiple protein present in the body. It also in the formation of all the cell organism in the body. So chromosomes is very important. Why? Because the formation of DNA also very important. So all the cell organelles we have to learn. It is very interesting, and we have to know about the cell organism and the type of all the cells. Thank you.